So in the previous video we had started with mechanism of action of fluorides. So far we have discussed two mechanisms. The first mechanism is increased enamel resistance or reduced enamel solubility. And the second mechanism is increased rate of post-eruptive maturation. So also just make a correction in the previous video. There was a spelling mistake. So this is ideal age of application. Okay, so when we talk about increased rate of post-eruptive maturation, the ideal age of application of the topical fluorides is 3, 7, 11 and 13 years. So the next mechanism is called remineralization of incipient lesion. Now what do you mean by incipient lesion? If you see this photograph, this white area, this is called as the white spot lesion. This is considered as the first sign of dental caries. Okay, so the first sign of dental caries is usually a change in the translucency of enamel. It starts becoming more whiter in color. This is a common situation you see whenever post-orthodontic treatment you remove the brackets, or a long-standing calculus you just remove. Below that you can see the areas of demineralization. So the first change, that is called as the white spot lesion, differs from the other caries lesion in that this stage is reversible. So when we talk about caries, it is not the brown color discoloration that we see first. So from this white lesion at this stage, if at all we are giving a proper remineralizing agent, you can remineralize the lesion. And beyond this, once it goes to the cavitation, it is irreversible. So when we talk about the definition of caries, we talk about it as an irreversible lesion. So that is beyond the stage. So this stage of white spot lesion or incipient lesion is completely reversible. So here what is happening is the mechanism is exactly what we discussed in the last mechanism of post-eruptive maturation. The only difference is that this takes place in a demineralized enamel and the previous mechanism takes place in a hypomineralized enamel. There is a difference between two. Hypomineralized enamel is an enamel where the mineralization is not yet complete, whereas demineralized enamel is an enamel where the mineralization is complete and it has undergone demineralization. So such kind of white spot lesion clearly indicates a risk of caries. And fluoride, when applied to these demineralized areas, which are also known as incipient lesions, over a period of time, they promotes the formation of fluorapatite crystals and remineralizes this white spot lesion. So that is the third mechanism. So the second and third mechanism are almost same, only that they occur in two different circumstances in two different time zones. That is the only difference between the second and third mechanisms. Ultimately, it is the fluorapatite crystal which is formed. The fourth mechanism is yet another systemic mechanism. Now, what is the meaning of systemic? I've also told you when we discussed about carbonate theory. This acts only systemically. That is, for this to act, fluoride has to be taken, ingested during the time tooth are developing. So similarly, research has shown that when fluoride is provided to the developing tooth, that is, again, you are systemically consuming fluoride and it reaches the developing tooth via the bloodstream. And when fluoride exposure is there to the developing tooth, what studies have shown that such a tooth, when it erupts into the oral cavity, it has shallow fissures and rounded cusps. So usually the most common type of caries you see is called pit and fissure caries. The reason is deep pits and fissures that can attract food debris. It is not accessible to your toothbrushes. This results in pit and fissure caries. But studies have shown that when optimum amount of fluoride is given during the time teeth are developing, the teeth which erupt later will have shallow cusps and shallow fissures and rounded cusps, thereby making it less susceptible to the caries lesion. And this is the tooth morphological alteration which fluoride can induce when given pre-eruptively. So this carbonate theory as well as alteration tooth morphology take place pre-eruptively before the teeth erupt into the oral cavity. For that, the delivery has to be through systemic route. The last mechanism, another important mechanism, is called interference with plaque microorganisms or caries causing microorganisms. Now, what you need to know in this is all the organisms. You are familiar with few of the organisms which cause caries, which include Streptococcus mutans or Lactobacillus acidophilus, to a certain extent Candida albicans or Actinomyces viscosus. All of this are acidogenic. The meaning of acidogenic is it is able to synthesize acids from carbohydrates. Now, where do these carbohydrates come from? The food you eat. So, the food you eat is fermented, or the carbohydrate is converted to lactic acid by a process called as anaerobic glycolysis. You all know, you must have heard, you have read in biochemistry that when glucose is converted to lactic acid, the procedure is called as anaerobic glycolysis. For aerobic, the end product is pyruvic acid. In the absence of oxygen, it gets converted to lactic acid. So exactly this is the mechanism taking place. Whatever carbohydrate food is consumed is converted into polymers of glucose, and this glucose undergoes anaerobic glycolysis by the ketogenic microorganism, resulting in production of lactic acid that causes demineralization. And this property of these microorganisms which are karyogenic is called as acidogenicity. All these organisms are also having the property of aciduricity. Now what is aciduric means is those organisms are able to survive in low pH. So when these organisms rapidly produce acids, the pH of the environment is going down or getting low, and the ability of these organisms to survive in that low pH is known as aciduricity. So all these karyogenic organisms are acidogenic and aciduric. Okay, so when we talk about this, I have already given the textual description of your mechanism of action in relation to interference with plaque microorganisms. You may go through that, but I'll be explaining that in relation to this diagram, which is easy to understand. All the textual part is also written there, but it is better to understand it using the diagram. So I'll be explaining this mechanism in relation to this diagram. So always the ionic fluoride if it is available as F minus, this is the bacterial cell wall. The fluoride does not enter the bacterial cell wall if it is in the ionic state. Okay. So when the pH of saliva, suppose this is the saliva environment, when the pH of saliva goes down, you all know when the pH goes down, the amount of H plus availability increases. Okay, so when the pH of saliva goes down, the H plus combines with F minus to form hydrofluoric acid, and that gets into the bacterial cell. And once it gets into the bacterial cell, it splits into H plus and F minus. So this is to what you should know is when the pH of saliva drops, that facilitates the entry of HF or the fluoride into the cell, and this effect is known as the fluoride pH effect. So fluoride pH effect is when the salivary pH falls, it increases the concentration of H plus that combines with F minus to form HF, and fluoride enters the cell. So entry of fluoride into the cell is facilitated by a drop in pH. This mechanism is known as fluoride pH effect. Now what happens is this H plus goes on accumulating within the cell. So if it goes accumulating beyond a point, it results in lowering of the intracellular pH, resulting in death of the cell. So always this H plus has to be constantly pumped out, and that is how this organism becomes aciduric. Okay, so this whatever H plus is getting inside is always constantly pumped out, and that is done by a ATPase pump. And fluoride inhibits this ATPase pump. This is the first effect of fluoride. It inhibits the ATPase pump. Next is glucose. So this, I told you, all these organisms are acidogenic. So it is able to synthesize lactic acid from glucose by a process called anaerobic glycolysis. So if you remember the glycolytic pathway, I am not enlisting all the steps. From glucose, one thing you need to know is there is a step called a conversion of 2-phosphoglycerate to phosphoenone pyruvate with the release of ATP. And phosphorol pyruvate is converted to pyruvic acid, that is converted to lactic acid. This lactic acid is secreted out of the cell. Now this conversion is enabled by an enzyme called as enolase. Okay, I just forgot to put this name. I shall correct it in the PowerPoint. So enolase is the enzyme which catalyzes the conversion of 2-phosphoglycerate to phosphoenone pyruvate. What happens is this fluoride inhibits this enolase enzyme. And because this enolase is inhibited, it does not result in the formation of lactic acid, and thereby it reduces the acidogenicity. So when fluoride inhibits the ATPase pump and reduces the efflux of H plus outside. It reduces the aciduricity. And when fluoride inhibits this enolase enzyme, it prevents the formation of lactic acid because this enolase enzyme does not work. So there is no formation of lactic acid. It reduces the acidogenicity. So this is one mechanism by which the aciduricity and acidogenicity is reduced. Next is glucose enters the cell through two mechanisms. The first mechanism is in an exchange of an ATP produced in conversion of phosphoenol pyruvate to pyruvic acid. That ATP which is produced is exchanged to glucose by a force known as proton motive force. PMF stands for proton motive force. Because this enolase is inhibited, this ATP is not generated and hence the entry of glucose by this mechanism stops. Similarly, another mechanism is called the phosphoenol pyruvate phosphate transferase system, which exchanges glucose to a molecule of ATP generated during this conversion. So because again enolase is inhibited, the entry of glucose by this mechanism is also stopped. So first fluoride stops the aciduricity, fluoride stops the acidogenicity, and fluoride stops the entry of glucose into the cell. Once this is inhibited, whatever glycogen reserve was there in the cell gets converted to glucose, but that is also a very limited stock. So when fluoride is present in high concentrations, all these mechanisms act together, resulting in the cell death. Or in high concentration, fluoride is bactericidal. At low concentration, it just inhibits the acidogenicity and aciduricity. It is bacteriostatic. So at low concentration, fluoride is bacteriostatic, and at high concentration, fluoride is bactericidal. Now this is what I have explained in all these steps. You can read that referring to this diagram so that you will be able to understand it easily. So these are the 5 mechanisms of action of fluoride in relation to dental caries. First is the increased enamel resistance or reduced enamel solubility. Second is increased rate of post-eruptive maturation of enamel. Third is remineralization of incipient lesions. Fourth is alteration in tooth morphology. And fifth is interference with caries or plaque microorganisms. So by that we come to the end of this topic on mechanism of action of fluorides and caries prevention. This is as I told you earlier, it is a long essay question. You need to understand all these mechanisms clearly in order to answer this long essay question. So that is all for today. Next week we'll continue with the topic on topical fluorides and systemic fluorides. Thank you.